Okay, so today I'm going to be presenting DISCO plus QR, rooting species trees in the presence of GDL and ILS. So first, a bit of context here. So what are species trees? So species trees are trees that sort of depict the evolutionary histories of species. So we have here the leaves, which are the species, and then the topology of the tree, which sort of represents the relationship in the evolutionary history. So for instance, here we can see that humans and chimps are more closely related than humans and gorillas. Now, one problem that we face when estimating species trees is that many species tree estimation methods actually return unrooted species trees. And the roots of the species trees are fairly important in determining how the evolutionary history of the species and how they evolved. So there's several different ways that people have come up to sort of root these species trees. So here's a couple more traditional approaches. So one approach might be outgroup rooting, which is when a set of species sort of more distantly related um, to the group of species than the one that's actually being studied is selected. And then when that's included in the species tree when they're estimating it, it gives an idea where the root could be. But the problem is that this can sometimes be difficult because it can be hard to select an outgroup because if it's too different, then it can be hard to figure out where to place it in the tree. And if it's too similar, then the root might not be clear. Another approach is distance-based methods such as merely rooting at the midpoint of the tree or other such methods. But the problem is this can be thrown off by deviations from a strict molecular clock. So this leads to sort of more new methods where we actually get signal from gene tree heterogeneity. Now, gene trees are trees that are estimated purely from one gene the as opposed to capturing the evolutionary history of an entire species. And often these gene trees might actually have differences between them and the species tree. And there's varieties of reasons for this. So one reason would be gene duplication and loss. So for instance, if a gene is duplicated, now there's two in the genome, and you could have a situation like this where we have a species tree of A, B, C, and D. Then there's a duplication event, and now we have a species tree with two A's, two B's, and two C's. And these two subtrees might evolve independently of each other. Um, and then another such source of gene tree heterogeneity would be incomplete lineage sorting. And this is a fairly complicated process, but it results in gene trees which have a different topology than the species tree. As we can see here, we have a species tree in sort of these blocks here, where D is separated from A, B, and C, and then gene tree where C and D and A and B are separate. Um, However, in this case, we do not end up with multiple copies of the same species in the gene tree. Um, so basically, the pipeline that we're looking at here is we want to have a set of gene trees, unrooted gene trees, and potentially multi-copy gene trees, so multiple copies of the same species, and then some unrooted species tree. And then we want to have some sort of method here that we can use all this information to get a rooted species tree. So there's several current methods that sort of approach this problem. So from different angles. So we have Stride, which uses GDL and multi-copy gene trees to infer the root, and then we have QR, which uses ILS in single copy trees to infer the root. However, QR is unable to handle multi-copy gene trees. And the problem with that is that often there's a lot of information in multi-copy gene trees, or like a large percentage of the input is multi-copy gene trees. So if you're restricting your analysis to only single copy trees, then you have to actually throw out a large quantity of the data, which could lead to lower accuracy. So this is sort of what we're addressing here. So we have this method called DISCO, and DISCO was originally developed to sort of basically enable species tree estimation methods that were designed for single copy trees to be used on multi-copy gene trees. And it does this by basically trying to figure out where the duplication events are in the multi-copy trees, and then splitting the trees apart into a bunch of single copy trees. And then these single copy trees can just be given to any method that uses single copy trees, and that pipeline should work. And it was shown that Astra Disco, which was Disco paired with a species tree estimation method called Astrid, actually was comparable in accuracy to Astral Pro, which was a leading method for multi-copy um, species tree estimation, but also significantly faster. So we can combine this idea, DISCO, with QR. So QR, for a little more detail here, uses ILS to root the species tree. It uses theory from Allman, Degen, and Rhodes from 2011, and it takes quintets in the gene trees, which are trees on 5 leaves, and then it looks at the distribution of the topology of these quintets, and it's able to, from that, sort of infer what the rooted tree should be. and and it was shown that QRLE, which is a faster version of QR, is at least as accurate as many different competing methods and can scale to large data sets. So basically, our idea here with DISCO plus QR is what we're going to do is we're going to decompose our multi-copy gene trees using DISCO and then use those as an input to QR. So, and sort of in pictures here, we have our set of multi-copy gene trees. We decompose them, and we have our set of decomposed trees. We have an unrooted species tree, which we could either have used a species tree estimation method to derive this tree from this set of decomposed trees, or we could get it from anywhere we want. But what matters is we have this set of decomposed gene trees in this unrooted species tree, and we can give both of these to QR, and at the end of the day, we should end up with a rooted species tree. So we decided to examine how well this worked on simulated data. So we simulated a bunch of model conditions, so varying from like 21 to 101 species with 1,000 genes for each model condition, and several levels of ILS. We had true and estimated gene trees. I mean, estimated species trees that we estimated with Astra Disco. And then we examined two different sort of pipelines here. So we just have the basic stride pipeline of we take the multi-copy gene trees, we give them to stride along with the estimated Astra Disco species tree, and then we get the rooting. And then we have our QR pipeline here, which is the same thing that we talked about. We decompose our gene trees. We get the set of single copy trees that we derived from the multi-copy gene trees. And then we take our estimated species tree and we get a rooted tree with QR. So here's just a brief overview of what we're going to see here. So basically, we're going to show varying rates of GDL and ILS. And when we vary the GDL, we find that stride tends to have an advantage when there's very high duplication rates, as sort of expected, since that's what it uses as its signal. However, when the duplication rates are low, it's very dependent on the ILS. So if there's a lot of ILS in the input, Disco plus QR is actually has a significant advantage over stride, and in that case. Um, and then when we vary the number of species, we see sort of similar trends here. So especially when there's lower numbers of species, the trends are similar, with Disco QR being better at high levels of ILS. However, at low ILS, and as the number of species increase, we find that there's sort of an interesting thing that occurs, which is that the differences between the methods seems to start to disappear with the increasing number of species. Um, anyway, so here is our results in more detail. So on this figure here, we have the y-axis, and it measures error in normalized clade distance. And now normalized clade distance is the percentage of incorrect clades in the estimated species tree that we got at the end of our pipeline, compared to a true tree that we have because we simulated the data. We have the exact true species tree. Um, and then down here on the x-axis, we have the duplication rates starting low on the left and going to high on the right. Um, so the light blue is stride, and we see that as the duplication rates increase, the amount of error that we have drops off pretty drastically. And once we reach fairly high levels of duplication here, we have so much signal that it does a fantastic job at finding the root. Um, Also, unsurprisingly, we see here in the darker blue DISCO+QR, it doesn't seem to be very affected in any way by the duplication rate here. It's just pretty consistent throughout. And this makes sense since it doesn't really use the duplication and loss as a signal in its finding of the root. So here we examine the high ILS case, and the sort of the trends of the methods in isolation are very similar here. We continue to see stride dropping off in or increasing in accuracy, dropping off in air as the duplication rates increase, and we sort of see a relatively consistent performance from DISCO+QR. However, the biggest note here is that the error is much lower overall for DISCO+QR, whereas there's maybe a slight increase in error for stride, as you can sort of see in a couple cases. And this leads to situations where under these low to medium duplication rates here, there's a very significant improvement that we're getting from DISCO+QR. Um, yep. So next we sort of examine the varying number of species. So when we vary the number of species, we sort of see here low ILS in 20 species, which in our previous slide we were looking at 21 species. So a very similar condition. And we see that, unsurprisingly, low ILS stride is slightly better since DISCO+QR doesn't have as much signal. However, slightly more surprisingly, as the number of species here increases, we see that the differences basically disappear. And especially here, there's virtually no difference whatsoever that's visible. um, and then here at the high ILS, we start out with DISCO+QR having the advantage as we have a lot of signal, and this advantage is maintained as the number of species increases. So anyways, the summary is basically stride becomes more accurate as the duplication rate increases, and DISCO+QR becomes more accurate as the ILS rate increases. So um, there's basically this trade-off. So um, the choice of what sort of method you want to use really does depend on the properties of your data set. So like how much ILS, how much GDL, and the number of species. And we do note that there is sort of this interesting impact of the number of species that could probably be further explored. In additional future work, we note that there's a lot of room for exploration as far as QR-based approaches. QR is a relatively new method, so it's sort of there's a lot of space here that's unexplored. Also, we note that examining against midpoint routing or outgroup routing might be a good idea. These are very common techniques that are more traditionally used. So having an idea how we compare against those might be good. And then exploring results from biological data might also be beneficial e- to get a better idea of how the methods work in practice. So anyways, thank you to our funding and my co-authors, and thank you all for listening.
Well, thank you. I'm going to start with my questions while I wait for something else. So have you tried different simulations with bacteria and eukarya? Because I think in some special cases in bacteria, for example, when a family is expanded, a family for a conserved family from primary metabolism, there is not a lot of lineage lost. And there's sometimes it's not extra copies by gene duplication, but by horizontal gene transfer. And maybe this is an ideal case for DISCO+QR. I just want you to answer if I'm interpreting correctly what your talk. And the other question is, if have you tried differences between bacteria and eukarya, and in which scenario your software tool works better?
Yeah, so we haven't like actually explored this that thoroughly. So I do think I know what you're talking about. So, like, I think in the Stride paper where it was introduced, they were showing that it was having a fair amount of difficulty on that kind of a data set, because it wasn't able to basically extract information because of the lack of sort of gene duplication and loss. And I think it would be very interesting to examine QR on that. A QR doesn't use horizontal gene transfer. It would be interesting to try to develop a routing method that integrates that idea as well. Yeah, and it would be yeah, I'm not super familiar with the bacteria data set, so I'm not sure how much ILS it has. But yeah, I really appreciate that idea, and I think that would be a good idea to explore, especially when looking at more biological data and such. Thank you.
Okay. Well, if there are not more questions, please, Alejandro, present our next speaker.
There's one.
Oh, there is one question, and we still have 2 minutes.
Hello, your method is designed for large or small branch distances, or does it matters? And also, I'd like to ask if incomplete lineage sorting takes any effect when you are analyzing very large evolutionary distances, or it only matters at populations level.
Okay, yeah. So yeah, how QR works is that it uses the topology of quintets. So it's actually entirely topology based. So the distances don't have a great effect on it either way. So yeah, that's actually sort of possibly an advantage, that it might have over something like midpoint routing and such. Um, if the distances are just because it's not very sensitive to that kind of stuff because it's purely looking at the topology. And the same sort of applies to Stride as well, where it's looking at duplication events, so it doesn't really, I don't think, take the lengths of the branches into account as much.
Okay, thank you.